So the topic of this lecture is the basics of the single particle image processing. And this is a quite broad topic, as you can imagine. The idea today would be just to describe the single particle pipeline. And I will try to explain you the basic ideas and what is happening when you are actually processing it, what is happening in the software behind just pressing the buttons. Even if you're not familiar with a single particle cryo-EM, I hope you would be able to follow this. And then, as always in real life, it's always very important to see the real life examples. And I will show you some work which we did during my postdoc with Paula da Fonseca at the LMB when we were working on the 20S parasite proteasomes. And since I'll be showing a lot about these proteasomes today, I just want to briefly introduce this complex, which is responsible for the degradation of other proteins within our cells. And this has been considered for many years as a test sample in cryo-EM. This is quite applicable for archaeal proteasomes, because archaeal proteasomes, they're composed of 2 alpha rings and 2 beta rings. And each of these rings contains 7 subunits, and they're all the same. All alphas are the same and all betas are the same. They behave very nicely on the grids, and that's why it's quite easy to test out some algorithms for the image processing and so on with archaeal proteasomes. But eukaryotic proteasomes, which I'll be talking about today, they're more challenging because all the alphas and all the betas are slightly different, which results in a pseudosymmetry, and also they don't behave so nicely on the EM grids, and that's a little bit of a pain. So, and before we start, I'm not sure, maybe this movie has been shown already today, but I hope not. So this is one of my favorites. It's from Gabe Lander from his PhD defense. The movie is quite old. It's more than 10 years old. And this shows the whole pipeline of cryo-EM data analysis. So we start with our sample, and we assume that it's purified, and the sample is, our protein is floating in the liquid in our buffer. And the idea is to get the structure of this protein. In this case, it's GRO-EL, by the way. So we take the grids, EM grids, which are quite small, and we apply a solution of the sample on the grids. You can see it here. And then what is happening in the Vitrobot, the excess of the buffer is being blotted away, and we have a very thin layer of the liquid, which is plunged frozen into the liquid ethane, which is cooled down to the temperature of the liquid nitrogen. So now when this procedure is happening, it's happening very fast, so that the ice is in a so-called condition which is called vitreous ice. And then all the molecules are fixed within the layer of this ice, and they're ready to go into the cryo-electron microscope. So now we can record images. We can record the projection images from the sample in the microscope, for example, like this. And nowadays, this is F20. Nowadays, we are using sometimes more advanced microscopes, but the idea is pretty much the same. So electrons are hitting with the beam, the particles, and this image is going to the detector, which can be film, or in the old days, or more sophisticated detectors like Falcons, or K2 or K3. And then we have the projection images. And this is how it looks like in the real life. The idea now, when we have something like that, would be to pick the particles. You can see here the side views and the top views of this GRO-EL. To pick them, basically, this is the selection of these molecular images, and then to you see they're all in different orientations. Now what we need is to align them with respect to each other, and then we want to get rid of the noise. We are doing denoising by averaging, and I'll focus on it a bit later. And then we have something which is called class averages. In the old days, especially, this idea was very popular to get the 2D class averages, and from the 2D class averages, we were going to the 3D. So basically, all the particles will get an assignment in 3D, and then as a final reconstruction, we would have something which would resemble the map, which would resemble the structure, hopefully resemble the structure of the object we are looking at. And 10 years ago, that was the resolution everyone was happy about. Now, pretty much that's not the case, because we are able to get a much higher resolution thanks to the so-called resolution revolution, which happened somewhat close to 2013, 14. And this has happened due to the major advances in the technological development of direct electron detectors, which Giuseppe was talking about. We have much better microscopes nowadays compared to 10 or 20 years ago, with much faster data acquisition. And instead of the operator pressing the buttons and going from one position to another, moving the stage, it's doing the whole procedure now automatically. It's very convenient, very fast. And with this, in parallel, the software has been significantly developing. We have now much better methodology, much better algorithms, and the computational resources are getting much lower in price. And this all together results in this such boom, which forced quite a lot of labs in the world who were doing structural biology to switch to from X-ray crystallography to cryo-EM, or at least to use it as a complementary technique. Unfortunately, cryo-EM is still not a routine for many samples. And again, I would like to point your attention and to say, what is cryo-EM limited by? And the main problem is the radiation damage. So if we would be able to expose the objects to very high doses, we won't have the problem that we would have very noisy images. But naturally, we are limited by that, and we need to deal with this very high noise levels. And the second thing which we need to remember is that the cameras, when they are recording the events, when they're registering events, when the electrons are hitting the camera, the chip, these events are often not recorded perfectly, and then we're losing there, and therefore the noise level being contributed by the cameras, productivity and the DQE and so on. And what's sometimes ignored, especially when you're just starting in the field, but this significantly important, is actually the purification of the sample. If you put some crap in, you'll get some crap out. And this is very fair for cryo-EM. The sample which is going into the microscope is of paramount importance. And sometimes you have issues with the heterogeneity. Sometimes you have issues with the behavior of the spaceman on the grid. And this all has to be tackled and optimized. So again, I started with the fact that we have our sample purified, and then it's ideal, but in most cases it's not. Very often one needs to deal a lot with the cross-linking, with detergents, optimize the buffer components, and so on. But let's assume for now that everything is fine. So the next thing which one's doing is usually negative staining. We need to figure out what would be the optimal concentration of the sample, how it would be behaving on the grids, to change sometimes the support to use graphene or carbon, or no support whatsoever. And then sometimes it's very important to treat the grids. And this is an example actually about the proteasomes I was talking about before, the 20 years. And 20 years from, in this case, it was a human proteasomes. And if you just put them on the negative staining, in negative staining on carbon, you would most likely see just the top views. And then you can apply some tricks like treatment with pentylamine. And then if you have pentylamine, you'll have top views, the side views, and then you would be able to get all the views possible to get the final 3D reconstruction. So the negative staining can be very important even already at this stage. But what's more important to do negative staining before cryo-EM optimizations is because sometimes the samples are quite cranky, and there are issues with the water-air interface. And during the procedure of the freezing, they can disassemble. And if you start analyzing your data set with something which is already fell apart, it's very hard to say what is happening with your sample. And sometimes negative staining might give you an idea about how a sample would look like before it's actually frozen. And then once everything is done and you optimize and you're happy with the concentration, how it behaves on low-level entry microscopes, then you're ready for the high-resolution data collection and processing. And it's very important to note that the very first data collection and the processing, if you have no idea about your sample, most likely will not succeed in high resolution reconstruction. And very often we need to come back to the biochemistry optimization. And this is very, very often the case. And in most of the labs, when we're doing cryo-EM, we repeat the cycle many, many times. So this is an iterative procedure, and you need to remember that coming back here might save you quite a lot of time rather than dealing a lot with the very bad data and trying to optimize and get high resolution reconstruction, just relying on the power of the single particles, particle analysis, or the methodology or the software which you're using. So this is the pipeline of the single particle image processing. You've heard today a lot already about the movie alignments. So we start with the movie alignments, then it goes to the CTF determination. Again, particle picking, we need to select the particles. We do the 2D classification. We need to figure out what are the side views, what are the top views, what are the intermediate views. Then from 2D, we need to go to 3D, and then we need to generate an initial model. All these steps I will describe later more in details, just to give you a basic idea of what's happening. And then we need to refine it. We need to refine, align these particles with respect to this reference, and hopefully we would get something like this. Most likely not, and then we would have to classify it further, either in 2D or in 3D. And we should go for 2D or 3D classification, and then finally, hopefully, we would get something which would be of significant resolution to interpret our data. And on top of that, we could do some steps like particle polishing and CTF refinement to get the very best out of your 3D reconstruction. So I will be talking a lot about denoising, about class averages. And if you're not familiar with that, it's very important to understand this. So what is happening? Let's imagine for simplicity that these are our particles. They are all in the same orientation. They are in very high, not very high, but quite high noise level. So you don't see all the details there. So the idea of single particle analysis is that when we have these noisy particles, we can actually average out the noise by, we can reduce the noise by averaging out the particles. And so what happens if we average out, if we create a class average out of these three particles? The sum of these three particles, sum or average, it would already be already contain much lower noise level compared to the individual particles. And then if we start to increase the number of particles, we would be getting better, much sharper and sharper image, because we would be reducing the noise and the contribution of the noise to this image. And this is called class average. So if we average out sufficient number of particles, we would get virtually noise-free class average. And this would be useful for going, for example, for 3D reconstruction or just analyzing in 2D. Now, when we're coming to the point of how are we doing and how practically we are implying all these algorithms, we're coming to the issue of the software. And in the very beginning, when you're just starting dealing with single particle, you realize that the software is very important, it takes quite a lot of time to learn. Actually, the software which we are using and for a number of years which we were using has been rapidly developing. It all started with these two packages, which are called Spider and Imagic. And these two packages were developed by Joachim Frank, who was responsible for Spider, and Marijn van Heel, who was responsible for Imagic. And these developments were going in parallel since the end of the 70s up to probably around 2010, when Reliance started to be taking over. These two packages were very popular in the past. There are some issues about this, and those are related to the ease of use. And it's quite difficult to use these two packages. It used to be quite difficult because they require really expert knowledge. And it was taking quite a lot of time to learn how to use it. And you need to know all the tiny details about the single particle analysis. But nowadays, with the development of the procedures and of the methods and so on, so we have very popular packages which are Reliant, CrySpark, and System. These three are probably the most popular. Sometimes people use XMIP quite a lot. And this is the modern software, and it's much more user-friendly. It's computationally faster, it's taking advantage of GPU, and nowadays it's rapidly developing, it's well-maintained, there are mailing lists and so on. You can always ask people around what is happening, and they might even write you scripts or to advise you to use the next edition of the software and so on. The truth behind this is that actually all the softwares, they're doing pretty much the same job. And it doesn't really matter which software you are using if you are doing your job well and you understand what are each stage in your analysis. What is more important is not just to press buttons, but to visualize your results at each step and to validate them. And I will explain you why here. So let's go step by step on the through the pipeline of the processing. And again, already it was told a lot about the movie mode data collection and analysis. And I probably could skip quite a few words here, but just briefly, we're collecting nowadays the data in the movie mode. And within the exposure, several frames are being acquired. And actually, you can see a movie of these particles which are moving within the ice layer due to different effects like beam-induced motion, it might be drift, might be charging effects, and so on. So the movie mode data collection allowed us to align the particles within the data acquisition, so we are getting much higher quality data after this procedure is applied. An interesting thing is that actually this has been described already in 1984, the whole idea of the movie alignments. And in 1984, obviously, all the data was usually acquired on the more higher resolution data if you want to. It used to be in cryo, it used to be acquired on the film because the cameras were slow and they were also quite not so sensitive in terms of their DQE. And in those days, this work was done on 256 by 256 camera, which and nowadays we have 4K by 4K. But the idea is pretty much the same. So what is happening? Let's assume we have 30 frames. And if we have 30 frames, we want to align them. We can split them into subsets, for example, into subsets of 5. And then when we average out 5 frames here, then we have this class average. And we can align each frame with respect to this sum. And then we can align each frame in the second subset here with respect to its sum or average. And then to repeat this procedure until convergence. And this is how actually the movie alignments is working. This is a global alignment when we are using the full micrographs, but you can also do patches. You can split them into smaller patches, or you can align them with already on the particles themselves in the later stages, which is called particle poaching. And also, movie mode data collection allows to compensate for the radiation damage, and this procedure is called dose weighting, something Christos was already talking about today. So once the movie alignments are performed, what is very important to do is actually to screen your data, especially if you've never seen what is happening on your micrographs before. And this is the danger of modern software packages sometimes, that actually you can start these downstream processing steps without having to check what was happening before. And if you skip the step of evaluation of how your movie alignments were done and what you have on the in the grids, you might end up in something quite nasty, and then you wouldn't be able to interpret your class averages later on. So if you bin your data, it's quite easy to screen to screen your micrographs and to detect something which was probably you might have different problems within the data collection since you're doing it automatically, and sometimes you might not get the full control about your ice thickness, and sometimes the micrographs can be taken on very dry areas, and there might be different types of contamination and so on. And you want to exclude it from further processing just because it will also save you quite a lot of time. So this is very, very important. Now, before we go to the CTF determination, and we will be talking a lot about Fourier space representation of the image data, I just want to, again, to focus your attention and to briefly remind you what it is. So this is a test image, so-called LENA, has been used for quite a while in different image processing techniques. And with any image, even a photograph, we can take as many Fourier transforms and to come back to the real space, back Fourier transform to create back Fourier transform or forward Fourier transform as many times as we want, and we won't be limited by the, we won't lose any information during these procedures. So the Fourier transforms, the amplitude spectrum would look something like that. It wouldn't remind us the real space. And this representation is very important for us because here, as you know, closer to the center of the image, we have lower frequency information, which is responsible for lower resolution details like the shapes of our objects. And this is important for the particle determination, everything which we will say in this circle. And when we are going further from the center of the image, and this is the Nyquist, by the way, and here, which is determined by your pixel size. And here we will be having the maximum possible resolution which can be obtained with this data set. So if we apply low pass filter, if we exclude all the high frequency information, low pass filter means that we are focusing on all the low frequency details, we would see an image which would look like this. We see only the shades and the general shape of the object. And if we are hiding all the low resolution information, we won't see any shades. We would see very crisp details. And this is the hair, for example, on this image. And this would be responsible for the resolution. Now, there's, again, we might be doing lots of different things, and during our analysis procedure, we might be doing binning. And this means actually that we are shifting the Nyquist towards closer to low resolution information. And this might be very important when you are at the initial steps of your processing, because there, for example, for the particle picking, you wouldn't need too much of the high frequency information. You just need the very low frequency details. And this is also fair for the alignments. Marta was talking about the CTF, and I probably don't need to talk too much about that. And in this case, we are talking about our sample again, coming back to the proteasomes. You can see here quite a poor concentration, but it was because we used the parasite sample and it was difficult to purify. We're comparing the experimental CTF with the theoretical values, and this allows us to determine the defocus within each micrographs was acquired at. And then later on, in those days, the CTF correction was done by phase flipping on the full micrographs. Nowadays, we are doing it during the refinement procedures on the particles and the isolated particles. That's why it's important to remember to have the box size of the particle quite large because of the localization of the CTF effects. I'll come back to this later. And again, Christos already showed a similar slide, but if you remember these particles from the original paper about the movie alignments, we can actually look at the power spectrum of the CTF to figure out some problems which are happening in our dataset, like this drift. And that's the alignment, so this information can be restored. Looking at the results of the CTF in a program like Reline or also in CryoSpark, it actually allows us to see the distribution of different, this histogram is a distribution of different defoci at which each micrograph was taken. It's important to check these things to make sure that you are not having all the data set which is acquired around the same defocus, because as Marta said, there are these zeros and we wouldn't have information in certain frequencies, and then in the enter CTF would look funny. So this is very important to check that your data was collected correctly, and then we can visualize the quality of the fit of the fit of our experimental data with respect to the theoretical CTFs. And if you see something like this, you can again select it and then exclude it from further processing. There are different criteria here, but I don't want to go into details. And typically, not all the micrographs would look like what I was showing to you before, where you can see nicely all the particles, but sometimes you see something like that. And that's actually quite also a good data set. It was also a proteasome data set, which was collected on a Titan Krice microscope with K3 detector, which was operating in counting mode. And why don't we see the particles? Again, as I said before, it can be due to the like our physical limitations in cryo-EM, that we're looking at the low defocus of the dose in the microscope and so on, the source of the electrons. But at the same time, it's important to remember that if you don't see something, it might be just because you have you're looking at the sample which would have particles in a very thick layer of ice, and then you would be limited by the contrast. The particle size is also very important. If they're too small, you won't be having a very high contrast unless you're using something like faceplate. Then the buffer components are very important. For example, if you have high concentrations of glycerol, it would be very difficult to pick the particles and then to align them and get a high resolution reconstruction and so on. Again, Marta had a similar figure here, and I really like this experiment from Fred Sigwarz. What it illustrates to us is the following. So this is the real data of a membrane protein of the ion channel. And here you can have the particle, which is isolated. And if you look at the projection from a 3.4 angstrom map, the particle would look like this. And this is the idea which we would like to have. And this is similar to what you were looking at when you had noise-free 2D class averages. But in real life, we have quite the opposite. Even if we reduce all the noise here, this is the simulated data, and if we reduce all the noise, we would have the effects of the CTF which are applied, and we also, well, we would be limited by the CTF. The image which you are looking here is quite far from what you would be willing to have. So therefore, this means that when we're looking at the cryo-EM data, we need to realize that we are actually looking at something which is quite far from the real objects that we want to detect. And we don't need to be fooled with that. Here comes one of my favorite topics. It's about the particle picking, because there are many stories about how particle picking can go wrong. So in the old days, again, particle picking was done by PhD students manually. And then sometimes you can imagine there are like several thousands of micrographs. A poor PhD student would be sitting and selecting all of them manually. That's unfortunately, that's quite sad, but that was the truth for many labs. Because the signal was so low that it was very difficult to detect with automatic procedures. And nowadays, it's quite different. And what can be done is to automate these procedures. It can be actually, there are several approaches, like automatic reference-free particle picking, where the particles are picked with respect to local variations within the signal within the micrographs, and basically we'll be picking everything which would be significantly different in the background in the micrograph, or automatic particle picking, which is usually template-based, and you're taking template from another data set, or you already process something and want to repeat, and then you're using internal knowledge about how the class averages would be looking like. Nowadays, it's very popular to use machine learning based approaches, and they are extremely good, very powerful, and they solve many problems which in the old days could not be solved. And two of them are just extremely good, like Topaz and Crayola are working very nicely. But again, one needs to remember that there are always issues with the particle picking. That these are two issues, of course. That there are false negatives. When you have poor contrast, you do not peak something or your objects which you want to pick. Some particles will remain unpicked. And there are also false positive peaks. And this is related to the reference bias quite often. And the reference bias is a very big thing. And this is the main danger in the whole field of the, or not the whole field, but in the whole pipeline of your image processing. You do not want to introduce into your dataset any reference bias. Why? There's a famous experiment, and this experiment is related to usage of like pure noise. So let's imagine that you have 1,000 pure noisy micrographs. So these micrographs are just computationally generated. They contain random white noise. So now let's take an image of Einstein, and we use this image as a reference, and we would be looking for particles in this data set which would look like Einstein's face. And always the machine, when you start correlating the face with the Einstein face with these micrographs, you would have some picks. You would pick something anyway. And you can then extract these particles. And if you extract these particles, you can apply the same approaches and the same algorithms I was talking to you about before. You can just average it out. And then what happens if you average out all these particles which are coming from this pure noise? And surprise, surprise, my keyboard doesn't work. But I guess you can already guess what is happening. Got stuck. That's Einstein face. And this is the class averages, the class average from all these particles which would be picked, and Einstein would be looking at you from something which does not contain Einstein at all. So, and this is a very big deal. And there was a very famous case. It's called Mao case, which happened in 2013. It's dedicated to the HIV trimer structure. And this protein first was obtained, the structure of this protein was obtained by tomography by Sriram Subramanian. And then in 2012, there was a structure from single particle from the group of Sadrowski and the postdoc in the lab, and the last name Mao. They obtained the structure of this HIV trimer with a resolution of close to 12 angstroms, as far as I remember. It was the dataset which was collected on a standard CCD camera on Polaris microscope with 600,000 particles. And 1 year later, using the same dataset, which they published the structure with, they actually refined the same dataset with the resolution of 6 angstroms. And this structure appeared to be very controversial. And there was a wave of very strange critics, which was related on the surface. For example, Richard Henderson was using quite strong wording about this, stating that probably there are no features of the particles in the data which Mao was picking whatsoever. And later on, there were 3 papers which appeared criticizing this paper as well, more in details. One was from Sriram Subramanian, one was from Maureen Van Hill, and again, a more detailed version from Richard Henderson, where they were explaining what was happening behind the scenes. And for example, here in this in the critics from Sriram, you can see on the left the image which was presented in this Mao paper. And he was showing the particles which he was picking. For some of you, that might remind something when we were looking at the Einstein's face, which was picked from the noise. And for comparison, he was showing his sample, which was acquired under similar conditions. And then it was raising the questions whether there are actually particles in this data set at all. And there was a long story, a long kind of fight between these guys. And in the end, Mao was forced to deposit the raw data. And actually, I looked at this data. It was deposited on the NPR archive. And it turned out that about 65% of the micrographs were completely unusable because he actually collected them probably in automatic fashion. I don't know how, but they were containing more carbon and ice than actual micrographs, which would be on the ice. And even those which were acquired on the ice areas did not seem to contain quite some material. And if to take into account the concentration which they presented in the paper, and then if to consider that all of these micrographs were supposed to be ideal, then the minimal concentration of the particles for picking would look something like this, not like this. This means that it's just impossible to pick 670,000 particles. The story pretty much didn't end up with anything, and the paper, both papers from Mao are not withdrawn. But later on, there were quite a few papers which were appearing with the same structure. They were in agreement with each other, but they were quite different from the structure which was published by that lab. So this story tells us that one should be very, very careful when introducing anything which might have any reference bias impact on the data which you are processing. And if you are using any kind of external knowledge, you need to verify that your data and your final structure is not biased by that. Now, let's assume that everything in the particle picking stage was done well, and then no reference bias was introduced. What is happening next? Next, we need to cut out the particles and to actually get rid of the noise and to detect the angles at which they are oriented in the space with respect to each other and get the 3D reconstruction. Sounds quite simple, probably, but what is happening behind the scenes when we're extracting the particles? First, we need to, when we're doing this cutting, cropping out these particles from the micrographs, we need to remember about the box size. And the box size usually has to be somewhat twice the longest diameter or the longest dimension of these particles. Once we have this, next the contrast has to be normalized. And this means that we need to have an average density of 0, and sigma has to be set to 1. And then, depending on the stage of the processing, depending on your magnification, particles can be binned. This is especially useful in the initial stages of the refinements or the centering procedures, the 2D classification, and so on. And then we need to invert contrast so that we have particles which would appear white on the dark background. And this is the standard for single particle cryo. Now, when we are doing this, we need to make sure that the particles, when we pick the particles, they are somewhat centered quite okay. It might be a tricky case for the elongated objects like this. We need to make sure that the box size, especially if we are extracting them for the 3D reconstruction, is sufficient enough to perform adequate CTF correction. And as I said, it has to be quite large. And then we need to check that the binning is not too large or not too small. It has to be always a compromise. And if when we pick the particles, we have more junk than the particles, we also should consider probably using different parameters for the particle picking. Now, you all remember probably this image I was showing to you before. So we are averaging out all these particles and we get this nice class average. Now let's complicate the task. And here you can see that the proteasome is looking at us with the side view and in the same orientation. But what happens if now we have the proteasomes which would be rotated and shifted randomly, and they would be looking at us like that? We cannot apply just an averaging like we had before, because if we just average 3 images like that, they are not aligned with respect to each other, and then averaging out everything will not give us any significant information. We would have something which would be the total average in case of like a sum of 1,000 particles would resemble the size of this object, and it would resemble a rotational average of each of those. So now this means that we need to find their orientations, how they are oriented in ice with respect to each other in 2D. This means that there are 3 unknowns here. We don't know their shifts with respect to each other, and we don't know the rotation angle. Now, if the particle is not centered, Then we need to find first these shifts, and this is done by the cross correlation. We can correlate the template, like this image 1 with this image 2, and then we would find that the shift would have to be performed to the point where we would have the highest cross correlation value. And then if it's rotated, then the rotational alignment has to be performed, and usually it's performed in the polar coordinates. So just we can imagine that each image can be represented in polar coordinates, and then we would have to find the value for these shifts. And basically these two would give us, these two alignments would give us an idea about, or not the idea, but would give us a way how to align them with respect to each other. So what is happening when we start to align them with respect to each other? Then we're going to the procedure which is called 2D classification. And nowadays, 2D classification implies that we are doing alignments when we're doing this classification in most of the softwares. So what is happening? Let's say that we would like to have 8 different classes. And then we can imagine that we can split again our full data set into 8 groups. And then each of these groups, we can take an average of all these particles in the same group. And then you would have something I was showing in the previous slide. You would have an image which would be which would resemble the size of the proteasomes. And that would be the iteration 1. At the next iteration, we would compare each particle with respect to each of these references. And then we would say that some particles might correspond, have high cross-correlation value towards certain references. And then that means that it would belong to the certain class. Then already on the third iteration, we would see that the particles will start to split between this circular view and this barrel-shaped-like view at low resolution still. And then we can repeat the procedure and use these as references again for the alignments of these particles. And then in the end, we would have something which would be already of much higher resolution with much finer details, which can be seen in these class averages. On top of that, in these classification approaches, it's important to note that instead of, so we're talking here about the probabilities of each particle belonging to a certain class. So if it's the top view, for example, it would have quite high probability to belong to any of the 4 classes and lower probability for belonging here. And this approach is called, you might hear about it, maximum likelihood. So as a result of this procedure, we would have all the shifts and all the rotational angles with probabilities to belong to the certain class average. And now when we have all these class averages, we need to go from 2D to 3D. And I won't stop too much here, and it's quite complicated, but this is the central section theorem, and usually it's introduced when we are talking about this thing, and it was first used in electron microscopy of biological objects in 1968 by Dorosia and Klug. So what is it about? So let's assume we have a structure. If we have a structure, for example, of our proteasome, we, in the microscope, in the transmission electron microscope, we would have particles which would result as the projections of this image into certain directions. And we know these directions, let's assume. And then it turns out that if we take a Fourier transform of each of these particles, it would be it would correspond to the central section of the Fourier transform, the 3D Fourier transform of this object. This means actually that if we know all these directions, we can reconstruct the 3D object, which would resemble our structure. If you didn't quite understand what it means, it might be easier to explain if we talk about in terms of tomography. In tomography, all the projections are acquired with known angles because we are tilting our sample. And in single particle, we are doing somewhat which is the reverse way. It's called back projection method for getting the 3D reconstructions. When we know these angles, we can get this 3D object. So it's just a reverse operation. So this means that we have two more unknowns, and which are corresponding to the angles on which would orient each particle in the 3D space. And this is out of plane rotation and the rotation around Z. Same as for the planes. Yeah, so in total we have 5 different unknowns which we want to determine. And once we determine them, for each of the single particle image, molecular image, we would be able to get a 3D reconstruction. And that brings us to so-called projection matching algorithm. So let's assume for now that we know about how roughly the structure should look like. And this is, again, against the rules because now we are using the reference, which is external, but let's assume for the simplicity for now, just understand how it works. So if we know how our reconstruction would look like this proteasome, which would be at a lower resolution, we can create forward projections from this reconstruction, from this 3D map reference, if you like. And then we would get, if we create forward projections in different directions, we would get a set of forward projections which can be used as references for alignment of each of these particles. And if we align each of these particles with respect to something which would have the highest correlation value, the highest reference which it would like, each particle, then we would get the angles. And this means that each of the particles would obtain certain angles which would correspond to their spatial orientation in 3D space. And then if we can create a back projection with these new angles, we will get something which would be of higher resolution because the angles would be more precise. And then we can create another forward projections from this new object, which would look much better than before. And these, again, can be used as references, and this whole procedure can be repeated until the convergence. And this is the whole idea about the refinements. Though, as I mentioned before, we need to be very careful because we can introduce the reference bias. And at some point, if we start to use references which would be too sharp, they might introduce so-called overfitting. And the noise would be refined against to certain features. And in the end, we might get something which would look like a hedgehog, if you like. And there will be some very sharp strikes coming from the center to the periphery. And this is a clear case of overfitting. And the softwares are dealing with that in their alignment procedures to limit the higher resolution information in the references. I don't have too much time to go into details about that. But so what do we need to know? Again, this is a chicken and egg problem. And we were talking that we take the reconstruction, which we already know, but how do we know that? And there are different ways how to create an initial 3D reconstructions. And I skip this first one using an external reference because it's dangerous. What the current softwares, modern softwares, are allowing us to do is use ab initio methods, and CrySpark and Reliant has a few algorithms for doing that, and it works quite nicely. So it actually takes advantage of the data within your data set itself. So in principle, you can think about it as you can randomly assign angles and then to hope that in these circles of projection matching approaches, you will get finally a nice 3D reconstruction. That's actually how it was working with angular reconstitution, which was implemented in Spider and the Magic. When the class averages were used, they were not so noisy, and with random startup, one might be able to get an initial 3D reconstruction. Or one can use the direct methods like tomography or random conical till to get an initial 3D reconstruction. One needs to remember that this initial model, as you could see, it's prone to be giving some errors during the refinement procedures, and therefore 3D refinements are very sensitive to the initial 3D reconstruction. One should remember about it very carefully. So how do we evaluate that the final result is correct? So if we split our full data set in two halves randomly, we can actually refine and apply this projection matching procedure, for example, to each of this half independently from another. And then we would have in the end, after the whole procedure converges, so-called half maps. Half maps because we split our data set into halves. And we will have two maps. What can we do? We can create a Fourier transform of one map and then a Fourier transform of another map, and to compare them in Fourier space. And this is called Fourier shell correlation. This is a bit complicated if you hear about it for the first time, but I'll try to explain what it means simpler. So Fourier shell correlation is a 3D function. But let's go one step back and we talk about 2D objects. So for example, for simplicity, instead of our particles, let's assume that we are comparing these two objects, one truck and three trucks, two photographs. They're quite different, and their Fourier transform would be quite different. So what can we do? We can, in Fourier space, take the shells, many different shells from the origin of the image. Then the first shell would be here, then there would be a few shells here, and this is probably the third or the fourth shell, and so on until we go to the Nyquist. And then what we can do, we can correlate the values within these shells and to build the so-called Fourier ring correlation plot. And if these two objects are independent and they have nothing to do with each other, they will not correlate, and the average correlation will be 0. Ignore this for now. Now let's come back. Yes, and this is the Fourier shell correlation, which was introduced quite a while ago. Now let's compare the same object as you see in the previous slide with a very noisy one. And again, if we take a Fourier transform of this one and of this one, we would have two different Fourier transforms. But here you can see that there is almost no signal which is coming in the higher frequency space. So when we construct the Fourier ring correlation here, we would see that actually, I did the slide in the morning, sorry, and it's supposed to be going to 0, but you can see that the correlation here would be positive. And this value, which is called half bit, or it's kind of a cutoff, it would be telling us at which stage the noise would be, the signal would be significantly higher with respect to the noise. And where this threshold would be crossing this this curve, the correlation curve, then we would be able to state that at this level, these correlations are significant with respect to the noise. And this is what the resolution is about in 3D. So now when we go to the 3D object, there are lots of debates about which Fourier shell correlation criteria to use. It doesn't really matter for now. For the didactical purposes, you should just realize that everything which would be higher than this noise values and which are determined by this cutoff values would be actually considered as the correlation which is sufficient to state certain resolution in our maps. What's more important actually in the end of the day is to look at your final map and to see the features which you're supposed to see to discover the biology which is behind and to answer your biological question. I'm finishing very soon. Just a few slides. I just wanted to show you the 3D refinements procedure of the data set I was showing to you all the time here, and this is the proteasome, this parasite proteasome. And it's here you can see the cycles of the 3D refinement when we're going from iteration 1 to the final iteration, and then you can see that it's refined to a very nice resolution, about 3 angstrom, and we're going into the one of the active sites, and then we can even locate the ligand here in red, which is bound to one of the active sites. Now, when you are at this stage, usually you could do even the local resolution measurements. There are some softwares like ResMap, which would probably provide you very nice plot, which would be lying to you most likely, because it would say that your resolution is closer to 2 rather to 3 angstrom as compared to before. But then you'll see very nice features, and this is pretty much ready for publication, right? But actually it's not. What is important here already at this stage is start to look at the problematic areas and to challenge what you are doing, what has been going wrong. So we were interested in different binding areas, the different active sites. As you remember, there was a ligand which was bound to one of the active sites, but it was a partial occupancy, which you can see there was a discontinuous density at another site. So this means that some particles might have the ligand bound and some particles might not have the ligand bound. Or there could be an option that ligand would be quite flexible, and this is a poorly clear indication of the poorly resolved area. So what do we do next? We need to classify out, and we need to perform 3D classification to split this into different states and to assign which particles would have the ligand bound and which particles will not have the ligand bound. This procedure is very similar to 3D refinement. And the difference is that instead of one reference, we would be using multiple references, which would be our 3D reconstructions. And it's very similar procedure, just instead of one set of projections, you would be having multiple, and this is also called multi-reference alignments. And in the end, you might, if you do everything correctly, you might get different classes, and these classes might represent different states. And in our case, to our surprise, we were working with the parasite proteasomes. We figured out that actually that was not a parasite proteasome in our sample, but it was a human proteasome. 50% of the particles corresponding to the human proteasomes and 25% actually corresponded to the parasite. The structures are very homologous and the sequence is very similar. Some areas are slightly different, and this difference can be used for this kind of classification. Why did it happen? Because when we were purifying the 20S proteasomes, this purification was done from the parasites which were grown in human cells, and in human cells, we had the contamination of the human proteasomes. So this also shows the power of the single particle analysis and the intrinsic power of single particle analysis, and also as a diagnostic tool. And it shows us that we should not really rely on everything which looks like very nice results ready for publication. We need always to challenge our results. So 3D classification is very, very complicated, can be very complicated. We need to split the compositional conformational heterogeneity, and there are multiple approaches. This is a very nice example. It was from Kyoshi Nagai's lab, where they were working on the spliceosome, and you can see here that the algorithm or the pipeline for this classification is very sophisticated. They were using different masks. They were using different types of classifications, so signal subtraction, many different things. I don't have too much time to go into details about that. It's quite an advanced topic. But one can consider going there when you're already at the stage when you have to deal with this heterogeneity. So further steps would be CTF refinement to compensate for CTF effects which were not compensated before, for the defocus, for the beam tilt to refine these values. Particle polishing can be done, and this implies beam-induced motion correction. Post-processing is just masking and filtering, and finally you need to validate the results, to check the resolution, to look at the hardness is correct, because in single particle we have 50-50 chance of getting different hardness. And just the very final slides, the building pipeline. So in the old days, I had to use different softwares for different steps. Nowadays, it doesn't really matter which software you're using, as long as you're getting good results and reliable results. So, and many people are using this very popular approaches across Spark, Reliant, System. Whatever works is good, as long as you take care of the results. And once there is reliable pipeline is established, you can apply it for any mutants or screening of the different substrates or the drugs and so on. That's the power of the single particle cryo-EM. And if you consider doing absolutely automatically on-the-fly data processing, you should remember that it's cool when you can, by the end of the data collection, get the movie alignments and CTF determination on all your particle picks. But you should realize that in this automatic procedure, you are accumulating more and more problems when you've gone downstream the single particle pipeline, and you might accumulate quite a few errors, and you need to take it with a pinch of salt. So take-home messages. I hope I convinced you that the single particle analysis is a very iterative process and requires lots of optimization. You need really to focus first on your sample purification, sample preparation, and then data collection and image processing would be valid if everything is okay at the previous steps. There are many softwares and there are many packages, and none of them currently is the best, and they often work complementary, and you should explore all of them and then to realize what actually your results are. And actually, your single particle pipeline can be tailored to your sample, and some software might work better than others. And just keep an eye on the result at each step so that you don't fool yourself and the referees. And with that, thank you for your attention.